Okay. Okay, maybe for a start, good morning, everyone. Thank you for joining us here in our guest lecture session. Today, here we have Dr. Shohei Uchinomiya. He is an assistant professor from Kyushu University. He is my very dear colleague and also my dear friend back in Singapore until now. So he's a very, very kind person. He has agreed to give us a lecture, 2 sessions even, this week and next week. So thank you so much for that. So we will learn together in this lecture about the microscope and fluorescence imaging for understanding of small world. So we have here Dr. Asep as our HOD. Maybe Dr. Asep, please, maybe you want to open this section for us. The floor is yours.
Okay, thank you. Very pleasant good morning to each and every one of you. Thank you for being with us today to attend this valuable activity supported by our faculty mobility program, who are organized by Dr. Muhammad Ridwan as head of undergraduate study, uh, Dr. Bayu Ardiansa, and also to Dr. Samira. Uh, so, on behalf of Department of Chemistry, Faculty of Science and Mathematics, Universitas Indonesia, we would like to express our gratitude to Associate Professor Dr. Shohei Uchinomiya, who is this morning give his valuable time to sharing with us his knowledge experience in this area of research entitled Microscope and Fluorescence Imaging for Understanding of Small World. Actually, in our department, we have about 550 students, consist of undergraduate student, master student, and Ph.D. student. But today it's around about 42 students who are attending this seminar. So wish you all the participants have a wonderful time and a proper seminar. So thank you again to Dr. Shohei Uchinomiya. Thank you.
Thank you, Dr. Asep. I think without further ado, maybe we can start our lecture this morning. Dr. Shohei, the floor is yours. Thank you.
Okay. Okay.
You may share your screen.
Yeah. Please wait. Can you see my slide?
Yes, it's very clear.
Okay. Okay, please wait. Okay. So, good morning, everyone. I'm Shohei Uchinomiya in Kyushu University in Japan. So thank you very much for coming to this class. I heard now it is 8 a.m. in your country, so it's very early time, but I heard the first class in your university start this time. But anyway, so this is very early time. So thank you very much for coming to this class. And I initially would like to appreciate Dr. Samira and other organized committee members for this class for inviting me to this great class. So I'm a researcher who is working on chemical biology field, especially in fluorescence imaging. So have you ever heard chemical biology field, chemical biology? I think many undergraduate students have not heard about the chemical biology. So this is a fusion research field of chemistry and biology. So today, I'm going to talk about the microscope and fluorescence imaging for understanding of small world. So actually, I had other class. I had this class consists of several sessions. I mean, the other 2 or 3 researchers are also invited for this class, and I heard the researchers are from organic chemistry and hydrogen energy. And my class is totally different from them. So because I will talk about a bit of biology and biological and medicinal research field. Okay. Please wait. Okay. So this class consists of 2 days. In the first day, today, I will talk about the microscope and molecular imaging. In the second day, I will talk about the fluorescence imaging. So Google Translator says me that fluorescence Indonesian language of fluorescence imaging is this. I'm not sure about the pronunciation, but Samira, is this correct? Is this
Yes, I think that's
fluorescence
a very
imaging?
good translation. Yeah.
Okay, good. Yeah. Google Translator always says me the correct thing. Okay, so I will talk about the fluorescence imaging and its applications in biological and medicinal field. So by the way, so can you see this arrow icon? So actually, when I use pointer, this work very slow. So okay, so let me use this arrow icon. Okay, and so the main theme in this class is seeing is believing. Again, this may be correct in Indonesian word. So if we can see the things, it is easy to believe it. So today I will talk about how humans see small things that we cannot see by our naked eyes. Okay, so this world consists of many things that we cannot see by our naked eyes. Okay, let's suppose that we go to a lake. Okay, so you can see many fishes and small insects. So there are trees and maybe even you can see many birds. However, there are other important living things. So as you may know, this is plankton. Okay, so these plankton make oxygen, and fishes and insects eat planktons. It is quite difficult to see the plankton by our naked eyes. However, they are indeed there. So for understanding of ecosystem, we need to recognize the existence of these planktons that we can't see by our naked eyes. So this is the same in the case of our human body. So human body includes several organs, such as the brain and the heart. We can see these organs by our naked eyes. Of course, we need to cut our body, but anyway, so we can see these organs by our naked eyes because they are relatively big. So, however, these organs consist of many types of cells, even including some bacteria in intestines. And these cells consist of several types of organelles, such as a nucleus and mitochondria. And these organelles consist of many kinds of proteins, small molecules, and even ions. So, and it is quite, so we cannot see by our naked eye these cells and organelles and protein, small molecules, and ions. However, for understanding of the function of our body, we need to know the function of these things. So, yes. However, the resolution of our necktie is not enough for see these small things. We can see bee and may be able to see the plankton. Of course, we cannot see the detailed structure of planktons, but maybe we can see the something, some small thing is moving or something like that. So, however, it is quite hard to understand the detailed structure of planktons. And we can also see the single hair. However, it is mentioned that the resolution of our naked eye is limited in 0.1 millimeter. So we can never see the single cells and bacteria, virus, and such as DNA. So for observation of these small world, humans have developed microscope and imaging technologies. Today, I will briefly talk about the history of microscope and molecular imaging techniques. So the key item for magnifying the things is lens. Human found that lens can magnify the small things. So you can understand this very easily because the lens is usually used in glasses like this. And you might use magnifying glasses when you are kids or even now. And importantly, this lens is also used in microscope. So the theory of magnification of imaging with lens is explained in this slide. So lens has two focus point. And if you put sample inside the focus lens can make the magnified virtual image. And we can see this virtual image through this lens. So we can see the magnified things, sample. And this is the case of single lens. And human notice that when we use two lenses called as objective lens and eyepiece lens, we can see the higher magnified image. So in this system, the sample is put out of focus length. So objective lens make virtual image here. And if the virtual image inside the focus length of eyepiece lens, eyepiece lens can make the higher magnified virtual image. So like this, by using two lenses, we can see the higher magnified image. Okay, so someone might develop the microscope before them, but many references say that the Hans and Zacharias Janssen in Netherlands initially developed microscope in 1519. And their microscope was very, very simple. They just put the two lenses as shown here, and the magnification was just several times. However, note that, okay, I explained in the last slide for the principle of magnification with lens. However, this theory was not established at his age. So in this age, they just put the two lenses like this without any theory. However, this is the initial microscope that human being developed. In 17th century, Robert Hooke developed the microscope, which can provide a more magnified image. So this is a model of microscope that he made, and this microscope equips two lenses here and here, and we put the sample here and observe from here from the top. Okay, using this microscope, he observed many small things. This is a sketch of tree that he drew. And he also found the cells for the first time. So now we said in your biological textbooks, there are many types of cells, and we know our body consists of many types of cells. But he initially found the cell. So can you see these? Sorry, this is a sketch of cork. Cork is a kind of plant. This is a dried sample. However, as you can see, the cork consists of many small loops like this. So he named these loops as cell. This is a concept of cell. Okay, then, and in the same age, the late 17th century, Leeuwenhoek in Netherlands also developed magnified glasses. So this is a handy type of magnifying glass. However, this glass is very, very worked well, and magnification was reached at 200 times. And here he puts the sample here and observes the sample from here, from this side. And using this glass, it's a very important point. And he initially found the microorganisms for the first time. So this is very, very important discovery for science. And now he is called as the father of microbiology. So again, this is a quite very important finding because human can understand there are many small things in this world. However, the people in 17th century didn't think so. Because the people mentioned, okay, we can understand there are many small things. However, are they useful for us and our life? They asked so. So the microscope was not very important for their life. And the microscope had been just hobby for rich people. Okay. So, however, this situation changed in 19th century because of the higher evolution of microscope. So in the previous slide, I mentioned about the theory of the magnification with lens like this. Okay. So this theory was established by Carl Zeiss and App in Germany in 19th century. So before that, people have developed the microscope only just based on their experience. However, after establishment of the theory for magnification of lens, so the function of microscope was drastically improved. And now they established a company named as Carl Zeiss. And Carl Zeiss is one of the biggest company of microscope in even now. Okay? So the improvement of microscope contributed not only to science, but also our life, especially the medicinal field. In this age, infection disease such as corona and pest have been one of the biggest issues in our life. So in this age also, the COVID-19 also was a very big issue for our life. But in this age, there was not good medicine for these infections. So in this age, the people didn't know the cause of infection diseases. That's why they sometimes thought this is because of the devil or age of the god. However, scientifically, there should be the cause of infectional diseases. So the researchers found the cause of infection diseases using microscope. So for example, the Koch in Germany was one of the representative researchers in this field. So in this research, researchers isolated saliva or blood from infected patient, and they observed what were in the infection patient by a microscope. And the researchers found a certain fungus was in the infected people. For example, the cholera and plague and the other diseases also. So of course, these are not virus itself, but still this is an important discovery for science and medicinal field. Based on discovery, people have developed medicines for the infectious diseases. So people recognize that it is very important to observe the small world for understanding of the mechanism of things, including disease. So nowadays, many types of optical microscope have been developed. So for example, the phase contrast microscope and other types of microscope. And now these microscope can provide the images at single cell levels. So optical microscopes are very powerful tool, not only research field, but also clinical use. So this is because the doctors judge the disease of patient by directly seeing the sample from patient with microscope. Okay, so for example, the doctors isolated the of course, not all of the organs or the part of organ, and they observe the structural change of organs in diseases. Okay, this is not the sample from human, and this is a sample of mouse, and used in my experience actually, and in the this is a picture of an image of normal liver. However, in fatty liver, you can see the clear structure like this, and these are actually fat. So by seeing these sample, we can say this is okay the fatty liver, and can judge how to cure these diseases. Okay, so so you can understand the importance of microscope in our life. Okay, so seeing is believing. So our resolution, the resolution of our necktie is just 0.1 millimeter. However, optical microscope enables us to observe small things such as cell and bacteria, which is very useful not only science but also our life. Okay, so however, human want to know the smaller things, for example, the virus and DNA structure. However, it is quite difficult to observe these things by optical microscope. So this is because the resolution of optical microscope is not still enough for observing the smaller world. So this is a brief explanation of theory of resolution. Here is the samples, and here is the lens, and here is the images. If these images are totally distinguished, can be distinguished from other samples, we can say the resolution is high. However, if these images are not distinguished from each other very well, we cannot say this is a high resolution. So this the resolution can be explained by the Rayleigh equation. So I'm not a specialist of this field, so let me skip the detailed explanation of this equation. But the most important point is this equation includes the wavelength of light. So the resolution depends on the wavelength of light. So that's why the resolution of optical microscope, of course, use light for observing samples. The resolution. Sorry. Please wait. Okay. So the resolution of optical microscope is 0.22 micrometer. So this is not enough for observing the structure of virus and the smaller things. So, please wait. Okay. So for example, in the biological field, optical microscope can provide a single cell image like this. However, in your textbook, you may see the illustration of single cell like this. So this illustration tells us that the single cell consists of various organelles such as nucleus and Golgi body and mitochondria. However, we cannot observe these organelles by optical microscope. So how did researchers found and understand these organelles in single cells? So for this goal, human developed electron microscope. So this is an electron microscope. So electron is a negatively charged particle around the nucleus, so in atom. And again, I'm sorry, but I'm not a specialist of this field, so let me skip the history of this part. But briefly, J.J. Thomson discovers the electron. And importantly, de Broglie climbs the hypothesis of matter wavelengths. So this theory is electron is a particle, negatively charged particle, but at the same, this is a wave. And this theory was experimentally demonstrated at a later age, at later. So electron has particle nature and wave nature. So this is very important point because in last slide, I showed the Rayleigh equation, including the wavelengths of light. So because the electron has wave nature, researchers thought, if they use the electron for observation of things, they can see the smaller things than optical microscope. So indeed, wavelength of visible light is this, and the wavelength of electron is much, much shorter than light. That's why. So development of microscope with super resolution can be achieved by using electron. So based on this idea, electron microscope have been developed in 20th century. So there are two types of electron microscope. One is a transmission electron microscope called TEM, and the other is scanning electron microscope called SEM. So this is a brief explanation of TEM and SEM. And in TEM imaging, TEM detects the transmitted electron. So which means electron pass through the sample. On the other hand, the SEM detects the reflected electron. So that's why TEM can observe the internal structure of things. On the other hand, SEM can observe the surface, provides the surface information of things. And it is mentioned that STEM can provide a higher resolution. Okay, so now you can understand why researchers can find the organella which are much smaller than the single cell, and how to draw the illustration of a cell in a textbook. The researchers observed a single cell with electron microscope and found many types of organella. So this is the electron microscopic imaging of mitochondria. So clearly, you can see the structure of mitochondria. For example, the crystal structure and also outer and intermembrane. You can also see the length of you can also see the distance of outer and inner membranes. So like this electron microscopic imaging provides a detailed structure at single cells. So recently, the combination of electron microscopic imaging and imaging processing technology can provide the detailed structure of organelles and their contact. Okay, so for example, this report provides the information of organelle contact between ER and Golgi body. So ER releases a small particle, and this particle fused with a Golgi. So they observed, of course, the image of the electron microscope is a bit difficult to understand, but by using imaging processing technology, they can clearly distinguish these structures. So for example, this is a Golgi body, this is a Golgi body, and this is a ER. And this image was made by this EM image. Okay. So like this, okay, so for understanding of function of cell, it is quite important to observe the small part like this. And also, as I mentioned, optical microscope can't provide the information of the structure of virus. However, using electron microscopic imaging, we can also observe the structure of virus. This is the structure of COVID-19. And as you can see, there are many spike structure like this. Because this type of virus is like a crown, so this type is named as corona. Okay, you can also find you you may also find the image of bacteriophage in your textbook. So in your textbook, okay, so this is like an alien and a bit difficult to believe this structure when we don't see the structure directly. However, the researchers directly observe this structure by electron microscopic imaging. So you can now understand how important the understanding of the small world is. Also, electron microscopic imaging provides a virus infection with host cells at single cell. So this you can see many virus here on the surface of host cells. So like this, electron microscopic imaging tells us the detailed structure of things, which helps us to understand the mechanism of biological process. So I also want to introduce the X-ray structure analysis. So this is actually not a microscope, but a quite important technology for understanding of small world, especially the structure of proteins. So okay, so every protein has its own structure. However, we can never see the structure even with by electron microscope. Electron microscope. Okay? However, actually, the researchers have disclosed the protein structures. Sorry. Okay, by using this X-ray crystal structure analysis. In this method, protein crystal was irradiated with X-rays, and researchers detect the diffraction light data, like here, and based on this data, we can make the electron density map, which provides a detailed structure of protein like this. So using this technology, people can understand protein include many structures, for example, alpha helix and beta strand seed. So now researchers make a huge library of protein structure called as protein data bank and share the information of protein structure for research. Every researcher can access to this data bank and gets the information of the protein structure freely. So this technology provides, for example, conformational change of protein before after ligand binding. Okay, so for example, this is the open structure of this protein. The distance of two domains is 6.57 nanometer. And after binding with the calcium ion, the distance changed to 4.09 nanometer. This may be the very small structural change, but this is quite important for the function of protein. So we can understand how protein function is based on the structural change using this technique. So this technique also can provide the binding formation of the ligand. Okay, this is this is an example. This is a protein called as glutaminase 1 and its ligand. So this is the structure of glutaminase 1, and you can find something here. This is actually the ligand. So you can know how does the ligand interact with target protein. So this is a ligand, and this is a protein structure. And like this, so can you see this dashed line? So this is a hydrogen bond formation. So, and as you can see, and like this, you can understand how ligand bind with target protein. So this is very important for understanding the function and also drug development. Okay, so the understanding of structure is very important for understanding the function and drug development. If we do not have any information about the protein, we can know we can't know this information. But when we get the clear structure, we can conduct a functional image for example, when the ligand bind with the protein, the conformation of the protein changes like this. And we can also use this information for drug design. So in drug or the inhibitor usually inhibits the protein function by interacting with protein. However, sometimes the initial compound the affinity of the compound is not enough as drug. So we need to improve the affinity of ligand and protein. Of course, we can modify the structure of ligand based on, so for example, trial and error. But if we know the detailed information of the ligand binding to protein, we can easily design the ligand based on the binding information and improves the affinity for this for the protein. So this is okay. So this method is usually used for drug development. So I explained how to use microscope in the field in biology and medicinal research because I'm a researcher in the chemical biology field. However, the microscope is also very useful for other research fields. So for example, in chemistry, the microscope provides the structural information of compounds. Usually, we identify this structure by NMR. However, of course, this NMR can provide a very important information about the structure of the target compound, but this is okay, so-called the indirect method. However, when we use the electron microscope, so we can directly see the structure like this. So you can see the benzene ring or like this. So also we can observe the chemical reaction with microscope, which can provide the information about the intermediate of the chemical reactions. So this paper reports the reaction of two fluorines. So two fluorines. So in this reaction, so in this paper, they observe the clear structure of the intermediate. So we can directly know how this reaction goes. Okay, so again, so seeing is believing. So the resolution of our naked eye is not enough for seeing this small world. However, with optical microscope in hand, we can directly see we can directly get the information of the small world. So the microscope have contributed to development of biology and medicine by showing us the detailed structure of the things. So again, so this microscope can be applicable not only biology, medicine, but also chemistry and material science or other things. So actually almost every research field. So you can understand how important the microscope is. However, of course, there are some limitations in optical microscope and electron microscope. So for example, the microscopes can't distinguish the two or more things by color. And so this is quite important in the heterogeneous sample. So microscope can't selectively detect our target protein. So I will explain later. And the microscope also can't detect the function of protein, which is the activity. I will talk about this point in the day second in my class. And so this is the case of electron microscope, and electron microscope can't conduct live cell imaging. Okay, so so initially I want to explain of a staying technology for distinguish the target from other things include conditions. Okay, for example, let's suppose that there are a lot of a cell group including many cells. However, okay, so this cell group actually include some type of cells. For example, the cell A and cell B and cell C. And let's suppose that we want to detect the cell type A selectively. However, optical imaging and electron imaging provides the information of this cell group. So, okay, so this image is just one the cell group containing only one type of cell, but the real sample includes many types of cells. So, which means very heterogeneous. However, if you can selectively stain the type A, we can easily detect this type of cell. So the staining of target is quite important for better understanding of target selectively. So, okay, so for more understanding of this point, let's suppose that you are a doctor and you want to detect an infected virus. However, this cell, the rare sample is very heterogeneous and include many types of cells. So if you observe just the shape of cell, it is quite hard to find the infected cells inside in this group. So we need to stain the infected cells selectively. So for overcoming this point, the researchers also developed a staining method. So actually, we means that I and Dr. Samira is a part of the researcher in this part. So in this part, and she also develops a very, very nice methods for the staining of the target cells. Okay, so again, so researchers have developed not only microscope and staining experiment for selective detection of our target in small world. So one of the famous staining technique is maybe the gram staining. This is a method for staining bacteria. This technique was developed by Hans Gram in Denmark. In this method, the bacteria are stained with crystal violet iodide. This is the staining dye. And after ethanol wash, the bacteria was also stained by safranin, and the cells stained by the crystal violet iodide shows a blue color, and the cells shows stained with safranin shows a pink color. Okay, of course, you don't need to understand the detail of the mechanism, but like this, using this method, we can clearly distinguish the cell type like this. So this is very important basic point. So for example, by using this point, the doctor can identify the cell type, and which allows the doctors to decide which kind of antibacterial drugs they should use. Okay, so the same thing can be said in biological research. The optical and electron microscope can provide the information of cell and organelles. However, cell and organelles contain many types of proteins and small molecules and even ions. Okay, this illustration shows the intramolecular environment. Okay, so like this, you can see the mitochondria here, and mitochondria also include many types of proteins like this. This is a tubulin, and this is a nucleus, and there are a lot of proteins inside cells. However, okay, this is our target protein that we want to see. Using optical and electron microscope, we cannot selectively detect this target. So how we can know the function of this protein in inside cells and even body? So to overcoming this challenging, researchers have developed the method called as molecular imaging. Okay, so again, we want to detect a target protein or small molecules in intracellular crude conditions. Okay, so this is our target. So for this goal, there are two important points. So in molecular imaging. So one is at this target. So we to aim this target. And the other is how to stain. I mean how to detect. So this our target. So in molecular imaging, researchers used a molecule called as probe. So for selective detection of our target. So this probe consists of ligand part and the detection moiety. And the ligand means a compound that selectively interacts with target molecule. And here is the detection moiety. And okay, so this is our target. So ligand can selectively bind with our target, and we detect here. So that's why we can detect where target is by observing this detection moiety. So that's why this is indirect detection of small target molecules. So this molecular imaging has contributed to biology and even medical care. Okay, today I will briefly talk about the two molecular imaging methods. One is a fluorescence imaging, and the other is a positron emission topology. And I will talk Actually, I'm a researcher in the fluorescence imaging field, and next week I will talk about the detail of fluorescence imaging. But today I will briefly introduce the fluorescence imaging. Okay, so let's go to the fluorescence imaging part. Okay, so before talking about the fluorescence, let me explain the absorption and the color. Okay, so this is a mango. So do you know why the color of mango is orange? So it is because mango is mango. So maybe it's correct, but scientifically, the mango is orange because mango absorb colors except orange. So visible light include every color like this based on its wavelength, okay? And the mango observed colors except orange. So that's why mango is orange. This is not because mango is mango. So like this, so this means mango include various chemical compound that absorbs the color. So this is the explanation of absorption of light. So some organic compound can absorb certain wavelengths of light. So here is a light source, and after passing through the organic compound, we can see the light, which include this light. We can observe this light, but this light includes only several colors, okay? Several wavelengths, not the full wavelengths of every color. And chemically, the compound with conjugation bond can absorb the light. And if the organic compounds doesn't have conjugate bond, the compounds don't absorb any light. Okay? And importantly, some organic compounds show fluorescence upon irradiation light. So after irradiation with the light, the organic compounds absorb the light as energy, and they need to release the energy after excitation. Okay. So actually, I will talk about the detail of this mechanism in the next my class. But here, please just understand some compound release the fluorescence after irradiation. After irradiation with light. So here is an example of such compound. Okay, this is the compound called as anthracene. And when this compound is irradiated with light, without light, this compound shows no fluorescence. But when this compound irradiated with light, this compound shows fluorescence like this. So these compounds are called as fluorescent dye. And to date, a lot of fluorescent dye have been developed. So here is the structure of the fluorescence dye. And these fluorescence dyes covers a lot of almost every visible wavelength of visible light. And these fluorescent dyes are used in fluorescence imaging for biological research. I want to explain how to use this fluorescence imaging in biological research. Okay, so you can find this kind of illustration for explanation of, so for example, the cell signaling in your textbook of biology. So as you can see, this illustration includes many types of proteins. And importantly, so for example, okay, so this protein moves to a nucleus. Okay, So this protein moves to mitochondria. You can see many illustrations describing many such illustrations in your biological textbook. Okay. However, how they know, how the researchers know this dynamics of proteins? Okay. So we need to know it. So traditionally, and of course, this method is currently also very powerful method. And so immunofluorescence have been used for observation of intracellular proteins selectively. So in this method, antibody, this is a kind of protein that can specifically interact with target proteins. So antibodies are conjugated with fluorescent dyes. In my previous slide, I explained about the probe, which is used in molecular imaging. So probe consists of interaction part and detection part. In this molecule, the interaction part is antibody, and the detection part is a fluorescence dye. And this fluorescent dye conjugated antibody is added to cell. And we can observe several fluorescence spots. In each spot, the antibody interact with target protein selectively. So by observing this fluorescence is corresponding to this fluorescence. And by observing this fluorescence, we can clearly know where our target protein is. And we can also know the dynamics of protein by observing the cells upon stimulation with something. Okay, so this is a fluorescence, immunofluorescence imaging taken by myself. And this is a protein localized at mitochondria. Can you see some structure here? This is a mitochondria. Okay. However, in the case of this protein localized at peroxisome, which is a vesicle type of organ. So you can clearly see the difference of the localization of these proteins. And this analysis also conducted, so if this analysis is conducted after stimulation, or stress condition of the cell, so the protein may move to other localization inside cells. So like this, we can selectively observe our target protein inside cells. Okay, so this fluorescence technique, there are various fluorescence imaging methods. And I will talk about in the second day, second next week. And briefly, there are some, for example, the fluorescent probes, fluorescent proteins, and also the fluorescent probes, which is a probe based on organic compounds, also have been developed. Okay, I will talk about the next week, the tip. Okay, so I also want to talk about the positron emission topology next time. This is a very important technology for clinical use. Okay, so in this method, radioactive isotope, for example, the carbon-11 or fluoride-18 are used as a detection mode. So radioactive isotope spontaneously decompose, resulting in release of positron. This is a positively charged particle. And this positron meet electron somewhere, okay? And followed by annihilation and release the gamma ray. And researchers detect this gamma ray like this. Okay. And in PET imaging, compound including radioactive isotope is used as probe. This probe is administered to our body. And gamma ray from this probe is detected. So like this, okay, this is a detector. And this detector provides image information of the, okay, information of the target. So such as a cancer. This is the image, the picture of the cancer in brain. So here you can see the cancer clearly. Okay, so importantly, as shown here, this technique is already used for clinical use. For example, especially the cancer imaging in our body. Okay, so the patient is administered with PET probe. And this is a picture of PET imaging in clinical use. So this circle is a detector. And here is a patient, and the patient goes inside this circle. And we detected the gamma ray from the PET probe. Okay, and we can conduct the cancer imaging, and doctors can understand where the cancer is in the patient. However, this is not very easy because you can understand, okay, there is a question, one question, several questions, but anyways, one important question is how PET probe can selectively achieve the cancer. This is a quite important point in molecular imaging. Okay, so there are several methods. And one of the most famous one is the fluoride deoxyglucose. Here, okay, so here is a radioactive isotope. This is a glucose conjugated with radioactive isotope. And this probe can be achieved, can go to cancer based on this system. Okay, I will explain. And glycolysis, you may know the glycolysis, which is one of the metabolic pathway for generation of energy. And this glycolysis is highly enhanced in cancers for generation of ATP and biomolecules. So that's why cancer use glucose, more glucose than normal cells for rapid cell proliferation. Okay, so this deoxyglucose can spontaneously accumulate in cancer. So based on this principle, we can conduct the cancer imaging like this. So here you can know the cancer. Here is the cancer. Of course, we can see the high signal at the brain and the kidney, but we can see several strong signal here. So here is a cancer. So we can easily understand where this cancer is inside our body. And of course, antibodies are also used in PET imaging. This is an example of cancer imaging with antibody type PET probe. Antibody is conjugated with radioactive isotope and administered to our body. And we can see the clear image of cancer. Okay, so that's why the molecular imaging technique is already used for clinical use. Okay, so again, let's go back to this slide. So humans have developed microscopes for understanding of small world that we can't see by our naked eyes. Now you can understand how important it is to understand this small world. And nowadays, the molecular imaging technique also also have been developed for selective detection of our target. So the combination with microscope, this is a magnification, combination with microscope and molecular imaging technique for selective detection, we can understand the small world better. Okay, okay, so I'm going to finish my class in the first day. It's a bit earlier, so I'm sorry, but this is my last slide. So in the next week, I will explain the detail of fluorescence imaging and its application in biological and medicinal field. So my take-home message is seeing, believing. Okay, so this is my, I'm going to finish my class. Okay, thank you very much for listening. So pay attention. Thank you very much for paying attention. Okay.
Thank you so much, Dr. Shohei. What a comprehensive explanation about microscope and molecular imaging. Maybe I can act as a moderator here a little bit. So if any questions from the students, in case they are shy to ask you, please go ahead. Any questions from the floor? Anyone?
It's okay. It's okay. It's a useful this session. And actually, Japanese students never ask questions. but
They are too shy.
no. It's because actually I'm not sure about the major of these students. Are they learn chemistry or biology? Everything. Yeah, still everything. Okay.
They are mostly from chemistry. The
Okay,
background is
chemistry part. Okay,
yeah.
so okay, so
I see most of them are undergrad students. Some of
okay.
them master and PhD students.
Okay, good. And because my class is a bit different from the chemistry, and so it may be a bit difficult to ask a question. However, I would like you to understand how important it is to observe the small world. And especially, so I will talk about the fluorescence imaging in my next class. And chemistry also quite important for the development of fluorescent probes. So, Samira, so are there any laboratory in chemical biology field in your department?
Oh, that's a very good question. Unfortunately,
Oh, okay.
we don't really have a dedicated laboratory for chemical biology, especially for the bioimaging.
Okay.
Hopefully in the near future, we're planning to have one.
Okay, great. Okay. So okay. So anyway, so the chemistry also quite important point for this fluorescence imaging. So even now you learned chemistry, you may go to the field of chemical biology, especially after graduation or get after master degree. You may go to the or PhD degree. You may go abroad for getting a PhD degree or as a postdoc. So in that time, so I would like you to slightly change the field, research field. I think it may be better for getting the very global viewpoint for science.
Yeah, I agree with you. It's very good advice. Even though the background is chemistry, but we can also broaden our field, right?
Yeah. So now sorry, I'm talking. I only talking. I'm talking. But so the nowadays, so fusion of research area is quite important. So biology is not the research field only for biological researchers. Okay. So chemists also can go to the biological field. And indeed, there are many, many famous chemical biology researchers based on organic synthesis, actually. So the many researchers go to the chemical biology field from the organic chemistry. Okay, so even now you learned the chemistry, so please learn or get knowledge about other research field. This is quite important from this age, okay? From this age, in this age and in the near future.
Great. Yeah. Thank you so much, Dr. Shohei, for your nice words. Maybe a last chance if anybody has questions, or maybe Dr. Shohei can give assignment for you.
Mm.
No, no, no. I'm kidding. Oh, I see. Is someone
Yes.
there? Okay.
Samira.
Okay. I think there is a student wants to ask you something, Dr. Shohei. Is that okay?
Okay.
Okay. Good morning. My name is Dina.
Hi.
I want to ask about what if can molecular imaging get wrong target, and then how if the target is wrong to detect? Thank
Oh,
you.
sorry. Can I ask again? Sorry. Thank you for asking a question. And can I ask again? Sorry. Or you can type.
Mbak Dina, could you please rephrase again your questions?
Yeah. Are my voice clear?
Yes, I can
Okay.
hear. Yeah, I can hear you.
Okay. Can the molecular imaging get target is wrong, and then how if the target is wrong to detect about this protein or else?
How long? Sorry, is how
I think
long?
what she's trying to say, I think, is it any case that the biological target for the molecular imaging is wrong, is not the correct target, and what happened and how we can conquer that?
Oh,
The
you mean
the biological
so
target for the fluorescent probe, like if you want to target some disease, right, like cancer, but the target is not really the target for the cancer.
Yeah. This is a very good question. So how can I answer to this? It is very difficult question. So okay. So actually, there are several. Okay. So initially, we need to carefully select the target. In cancers, okay. So cancer. Okay. So cancer express the certain type of proteins. Okay. So we can okay. So how to ask? Okay. So do you know the okay. So anyway, initially we need to carefully select the target in the disease. Okay. So and as you mentioned, the actually fluorescent probes or antibody cannot selectively bind to our target. It is called as non-specific interaction. Okay? So in this case, we need to improve the selectivity of the antibody or the chemical probe. Chemical probe means the organic compound. So this is quite important point for the in this research area. Okay. So is okay?
So we have to improve selectivity.
Yes. The selectivity is very important. So for improvement of the selectivity, we can design the Okay, so design the molecule, the probes. And for example, in the case of fluorescent probe, the chemist can change the structure of the organic compound. Okay, so for selective, okay, so that's why. So we need to the knowledge of the chemistry for in this research area. Okay.
Okay, thank you.
Thank you, Dr. Shohei. I think we have another questions and in the chat. I think they are too shy
Yeah.
to ask you directly. Can I please read the question for you?
Okay, okay. So, okay, I have a question about the type of instrument that can determine the 3D protein structures in the sample. 3D structure of sample. Okay, X-ray crystal analysis can provide the 3D structure of samples. Okay, for example, I will show you. Please wait. 3D structure. Yeah, this is very important point. And actually, okay, so can you hear? Okay, so can you see this slide? Okay, this is a website of. Okay, I will show you. Okay, this is a website of PD protein data bank that I showed you. And here you can get the information of 3D structure of protein, for example, like this. Okay, so here is a ligand also, and you can understand how this. Okay, sorry, this is not a ligand but solvent. Okay, I will show you another one. Okay, can you see this? Okay, here is a ligand. Okay, oh, where is the ligand? Here. Okay, here. Here is a ligand, and you can also know how this ligand interact with protein like this. So you can get the 3D structure, the information of 3D structure using the X-ray crystal analysis. Okay, is it okay? So the other question is, how can we observe the interaction between the virus and the bacteria and with host cell under the microscope? Does it mean that to observe the cell interaction with the bacterial virus must be the living system? Okay, so initially, actually the okay, so I show the image of electron microscope. However, electron microscope cannot provide the live cell imaging. So that's why the fluorescence imaging for getting the live cell imaging have been developed. I will explain this point in my next class. And how can we observe the interaction? How can we observe the interaction between the bacteria and the host cell under microscope? Okay, so if this question doesn't mean for getting the binding contact, binding constant or some value, we can just see the interaction by optical microscope with microscope. We just see the interaction, how can I say the attachment of attachment. For getting other, so for example, the scientific value, we need to conduct another experiment. Is it okay?
I think Angelique Mirel. . I think that answers our question.
Okay, thank you very much for your question.
Maybe any more questions? Or otherwise, maybe Dr. Shohei, maybe you can conclude your class for today's class.
Okay, so thank you very much for coming to my class, and I'm very happy to meet with you. And okay, so next in my next class, I will talk about the fluorescence imaging, where the chemist can contribute. And I will explain how the power of chemistry have been contributed to this area. Okay, so see you in the next class. Thank you very much for coming to my class. Okay.
Thank you so much, Dr. Shohei. Everyone, we'll meet again next week.